Welcome, everyone. I'm excited to kick off our session today with an overview of Module 1, which is a general introduction to medical biochemistry. Today, we'll be focusing on Study Session 4, an introduction to nucleic acid biochemistry. This is part of BCHM 241, the biochemistry of macromolecules. Over the course of this presentation, we'll explore the fundamental aspects of nucleic acids, their structure, function, and significance in biochemistry. So, let's get started and dive into this fascinating topic. Let's dive into the chemistry of nucleic acids, which are essential molecules in all living organisms. First, we'll look at their functions. Nucleic acids are primarily responsible for storing and transmitting genetic information, which is fundamental to inheritance and cellular function. Understanding their components helps us grasp how they perform these roles. Next, we'll explore the components of nucleic acids themselves. These molecules are built from smaller units called nucleotides, which are the basic building blocks. Each nucleotide consists of a sugar, a phosphate group, and a nitrogenous base. Finally, we'll differentiate between nucleotides and nucleosides. Nucleotides include the phosphate group, making them the active units in genetic processes. Nucleosides, on the other hand, are just the sugar and the base, without the phosphate. Recognizing these distinctions is key to understanding the structure and function of nucleic acids in biological systems. Nucleic acids are organic molecules that contain important elements like carbon, hydrogen, oxygen, nitrogen, and phosphorus. These molecules were first discovered inside the nuclei of cells, which makes sense given their crucial role in genetics. Their primary function is to serve as repositories and transmitters of genetic information, ensuring that traits are passed from one generation to the next. Nucleic acids are long chains made up of repeating units called nucleotides, and these chains are known as polynucleotides. These chains are held together by phosphate bridges connecting the 3 and 5 ends, which is essential for their structure and function. There are two main types of nucleic acids, and they are chemically distinct: DNA, or deoxyribonucleic acid, and RNA, or ribonucleic acid. Both are vital for life, but they have different roles and structures, which we'll explore further as we go along. Understanding these molecules helps us grasp the foundation of genetic information and how it is stored and used within living organisms. Let's start with the discovery of DNA. In 1869, Johann Friedrich Miescher, a young Swiss researcher, identified a substance in the nucleus of cells that he called nuclein. He observed that this substance was acidic and quite different from other known molecules at the time. This was the first step toward understanding DNA's role in biology. Fast forward to 1944, when Avery, MacLeod, and Metcalfe demonstrated that DNA actually carries genetic information. This was a pivotal moment because it shifted the perception of DNA from just a chemical substance to the blueprint of life itself. Then, in 1953, James Watson and Francis Crick proposed the famous double helix model for DNA's structure. Their model was simple yet elegant, revealing how the strands of DNA twist around each other. This discovery was crucial because it explained how genetic information is stored and replicated. But their breakthrough didn't happen in isolation. It was built on the work of other scientists, notably Rosalind Franklin and Maurice Wilkins. They used X-ray diffraction techniques to study wet fibers of DNA. The diffraction patterns they obtained revealed key structural features, such as the helical shape, the presence of more than one strand, and about 10 base pairs per turn. These insights provided the physical framework that Watson and Crick used to build their model. We also have to mention Erwin Chargaff, whose rules about the proportion of bases in DNA, like the fact that adenine pairs with thymine and cytosine pairs with guanine, were critical for understanding the structure. Linus Pauling also contributed to the field with his work on molecular structures, although he focused more on proteins. In summary, the history of DNA's discovery is a fascinating story of scientific collaboration and incremental breakthroughs, all of which laid the foundation for our current understanding of genetics. Let's take a moment to explore the history behind this topic. Understanding the origins helps us appreciate how it has evolved over time. Throughout history, key developments have shaped the way we approach this subject today. These milestones highlight the progress made and the challenges faced along the way. By connecting these historical points to our current understanding, we can see how past innovations and lessons continue to influence our work now. So, as we move forward in this presentation, keep in mind how this history forms the foundation for the ideas and solutions we're discussing. Now, let's take a closer look at the fundamental building blocks of nucleic acids. Nucleic acids are made up of monomers called nucleotides. Each nucleotide is composed of three main components. First, there's the nitrogenous base, which you can think of as the informational part of the nucleotide. These bases are classified into two categories: purines, which have a double-ring structure, and pyrimidines, which have a single-ring structure. This distinction is crucial because it influences how nucleotides pair up in DNA and RNA. Next, we have the pentose sugar. Depending on the nucleic acid, this sugar can be either ribose in RNA or deoxyribose in DNA. The difference is just one oxygen atom, but it has a big impact on the molecule's properties and functions. Finally, there's the phosphate group, which links the nucleotides together to form the backbone of the nucleic acid strand. The phosphate can be attached as monophosphate, diphosphate, or triphosphate, depending on the number of phosphate groups. These variations are important because they relate to energy transfer and genetic information storage. All these components, nitrogenous bases, sugars, and phosphate groups, combine to form the nucleotides, which are the basic units of nucleic acids. Understanding these parts helps us comprehend how genetic information is stored, transmitted, and expressed. Let's take a closer look at the purines and pyrimidines rings in DNA and RNA. In DNA, we have four main bases: adenine, which we abbreviate as A, guanine, G, thymine, T, and cytosine. See now, RNA shares three of these bases: adenine, guanine, and cytosine. But instead of thymine, it contains uracil. It's important to note that adenine and guanine are classified as purines, which have a two-ring structure, while thymine and cytosine are pyrimidines, with a single-ring structure. In DNA, thymine is replaced by uracil in RNA, so RNA doesn't contain thymine at all. When drawing these purines, they are numbered in an anti-clockwise direction, while pyrimidines are numbered clockwise. This numbering helps us understand their structure better. Understanding these base structures is crucial because they form the foundation of the genetic code, and their specific arrangements and differences are what enable us to distinguish between DNA and RNA at a molecular level. This connection to the overall theme of genetic information is what makes these details so vital. Let's take a closer look at purine bases. These are fascinating structures because they have a 9-membered ring system that combines a pyrimidine ring fused to an imidazole ring. When we talk about the atoms within the purine ring, it's important to note that they are numbered in an anti-clockwise manner, which helps us precisely identify different positions on the ring. The major bases we find in nucleic acids are adenine and guanine. Adenine, or 6-aminopurine, has an amino group attached specifically at the 6th position. Guanine, on the other hand, is known as 2-amino-6-oxypurine because it contains an amino group at the 2nd position and an oxygen atom at the 6th position. Understanding these structural differences is crucial because they influence how these bases pair and interact within the DNA or RNA molecules. Recognizing the specific groups attached to the purine rings helps explain their chemical behavior and their role in genetic information storage. So, in summary, purines are characterized by their unique fused ring structure, with adenine and guanine being the key players in nucleic acids, each with distinctive functional groups that define their properties. Pyrimidines contain a 6-membered nitrogenous ring, which is fundamental to their structure. When we look at this ring, the atoms are numbered in a clockwise direction, making it easier to identify specific positions on the ring for different functional groups. The major pyrimidines we find in nucleic acids are cytosine, uracil, and thymine, each playing unique roles in genetic material. Let's start with cytosine. Cytosine is present in both RNA and DNA, which makes it quite versatile. Its chemical structure is called 2-oxy-4-aminopyrimidine, meaning it has an oxygen atom at position 2 and an amino group at position 4. These groups are crucial because they influence how cytosine pairs with other bases. Moving on to uracil, which is found exclusively in RNA. Its structure is known as 26-dioxypyrimidine, with oxygen atoms at positions 2 and 4. This specific arrangement is important because it affects how uracil interacts during RNA processes, especially in base pairing. Lastly, thymine is another pyrimidine found in DNA. It is similar to uracil, but has a methyl group attached at position 5, making it methyluracil. Thymine also has oxygens at positions 2 and 4. The presence of this methyl group helps distinguish DNA from RNA and contributes to the stability of the DNA molecule. Additionally, there are some unusual pyrimidines like methylcytosine, which is present in DNA, and dihydroracil, found in tRNA. These variants add further complexity and specificity to nucleic acid functions. Understanding these pyrimidines, their structures and differences, is key to grasping how genetic information is stored and transmitted at the molecular level. Today, we're going to explore modified pyrimidines, an important aspect of nucleic acid chemistry. These are essentially pyrimidine bases that have undergone chemical modifications, which can significantly impact their function. Such modifications often influence DNA stability, replication, and repair processes. They can occur naturally, for example, through DNA damage caused by environmental factors like UV light, or they can be introduced artificially for research or therapeutic purposes. Understanding these modifications is crucial because they can alter genetic information, sometimes leading to mutations or disease, but also offering potential avenues for targeted drug design. Throughout the presentation, you'll see how these modified bases fit into the bigger picture of genetic regulation and cellular function. So, grasping their chemistry and biological significance helps us better understand how genetic information is maintained and manipulated. Let's take a closer look at modified purines. These are interesting because they illustrate how purine structures can be altered to serve different biological functions. Modifications often occur in metabolic pathways and can influence how purines are utilized or broken down. Understanding these modifications helps us better grasp the complexity of nucleic acid chemistry and the regulation of nucleotide pools in cells. They also play a role in various diseases, including cancer and genetic disorders, making this a crucial area of study. So, recognizing how purines can be modified gives us insight into both normal cellular processes and potential therapeutic targets. Now, let's take a closer look at ribose and deoxyribose. These sugars are fundamental in distinguishing DNA from RNA. The key difference lies in the pentose sugar present in each molecule. DNA contains beta-D2-deoxyribose, which means it has a hydrogen atom at the 2 position instead of a hydroxyl group. On the other hand, RNA contains beta-D-ribose, which has that hydroxyl group at the 2 position. An important detail is how the atoms in the sugar are numbered from 1 to 5. This numbering helps us differentiate between the sugar and the nitrogenous base it's attached to, ensuring clarity in understanding their structure and function. So, in summary, the type of pentose sugar, whether it's ribose or deoxyribose, is what helps us tell apart DNA and RNA at a structural level. This distinction is crucial because it influences the stability and overall behavior of these nucleic acids within biological systems. Let's take a closer look at the phosphate group. This is a crucial component in many biological molecules, especially nucleotides and nucleic acids like DNA and RNA. The phosphate group consists of a phosphorus atom bonded to 4 oxygen atoms. It plays a key role in storing and transferring energy within cells, such as in the form of ATP. Additionally, phosphate groups help form the backbone of DNA and RNA, linking nucleotides together through phosphodiester bonds. Recognizing the importance of the phosphate group helps us understand how genetic information is maintained and how energy flows in biological systems. Overall, the phosphate group is fundamental to the structure and function of life at the molecular level. Let's talk about nucleosides. These are formed when a sugar molecule covalently bonds to a heterocyclic base. The key point here is that this bond is called a glycosidic bond, and it's crucial because it links the sugar to the base securely. Now, when we name these nucleosides, we add specific suffixes depending on the type of base. For pyrimidines, we add idine, so you get names like cytidine, thymidine, and uridine. For purines, we add osine, resulting in adenosine and guanosine. Understanding these nucleosides is fundamental because they form the building blocks of nucleotides, which are essential for genetic information storage and transfer. So, in summary, nucleosides are basically the first step in creating the larger molecules that carry our genetic code. Let's talk about how nucleosides are formed. Basically, when you combine a base like adenine with a sugar, you get a nucleoside. For example, if you take adenine and attach a ribose sugar, you get adenosine. If you attach a deoxyribose sugar instead, you get deoxyadenosine. This is really important because in DNA, the building blocks are deoxynucleosides. There are 4 main types: deoxyadenosine, deoxyguanosine, deoxythymidine, and deoxycytidine. These are the fundamental units that make up the structure of DNA. Understanding how nucleosides are formed helps us see how genetic information is stored and transferred. It's a simple concept, but a crucial part of the bigger picture of molecular biology. Nucleotides are the building blocks of nucleic acids, and they are formed through a specific process. Essentially, they are made by condensing a sugar, a phosphate group, and one of the 4 bases. When we talk about nucleotides, it's important to understand how they are built. They are formed by joining a phosphate group to a nucleoside, which is a sugar attached to a base, at either the C5 or the C3 position on the sugar. The bond that forms between the phosphate and the sugar in this process is called a phosphoester bond. So, in simple terms, a nucleotide is just a combination of a base, a sugar, and phosphate groups. For example, when we look at specific nucleotides like adenosine monophosphate, or AMP, adenosine diphosphate, or ADP, and adenosine triphosphate, or ATP, these are all just different forms depending on how many phosphate groups are attached. When naming nucleotides, we add the suffix -ylate to both pyrimidine and purine bases. This helps us identify the specific nucleotide. For pyrimidine nucleotides, we have examples like cytidylate, thymidylate, and uridylate. For purine nucleotides, the names are adenylate and guanylate. Understanding these structures is fundamental because they form the core of genetic information storage and transfer. They are essential for energy transfer, signaling, and many other biological processes. This slide doesn't contain enough information. That's why we're moving to the next slide. Polynucleotides are the building blocks of genetic material, and they play a crucial role in storing and transmitting biological information. When we talk about polynucleotides, we're referring to long chains made up of smaller units called nucleotides. These nucleotides are linked together through phosphodiester bonds, forming a backbone that is both stable and flexible. The most common polynucleotides you'll hear about are DNA and RNA. DNA, or deoxyribonucleic acid, contains the genetic instructions used in the development and functioning of all living organisms. RNA, or ribonucleic acid, is involved in protein synthesis and various other cellular processes. Understanding the structure and function of polynucleotides helps us grasp how genetic information is stored, copied, and expressed. This knowledge is fundamental to fields like genetics, molecular biology, and biotechnology, and it ties directly into the overall theme of understanding life's molecular basis. So, in essence, polynucleotides are at the core of life's blueprint. Let's take a closer look at cyclic nucleotides. These molecules are fundamental building blocks of DNA and RNA, serving as the genetic material in all living organisms. They are also crucial as carriers of chemical energy. For example, ATP is the major energy source used throughout the cell for various processes. Other nucleotides like GTP, UTP, and CTP also supply energy during specific metabolic pathways, helping to drive reactions forward. In addition, nucleotides are key components of enzyme cofactors, such as NAD, NADP, FAD, and coenzyme. These cofactors are essential for numerous enzymatic reactions, especially those involved in metabolism and energy production. Furthermore, nucleotides act as chemical messengers. For instance, CAMP functions as a second messenger inside the cell, transmitting signals from hormones and other signaling molecules to elicit specific cellular responses. Similarly, cGMP acts as a cellular mediator, modulating processes like vasodilation and sensory transduction. Understanding these roles highlights how nucleotides are not just the building blocks of genetic material, but also vital players in energy transfer, enzyme function, and cell signaling, integral to the overall functioning of life at the molecular level. Let's wrap up by exploring how DNA is packed inside the nucleus of a cell. You might imagine all that genetic information tightly coiled to fit into such a tiny space. In reality, DNA is wrapped around proteins called histones, forming structures known as nucleosomes. These nucleosomes further fold and loop to create the highly organized, compact structure of chromatin. This organization is crucial because it allows the cell to efficiently store and access genetic information when needed. Now, why do we often describe nucleotides with terms like acid and negative charge? Well, nucleotides, the building blocks of DNA, contain phosphate groups that are negatively charged. This negative charge is essential because it influences how DNA interacts with other molecules, including proteins and enzymes. The acidic nature of DNA helps maintain its stability and also plays a role in how it is recognized and processed during processes like replication and transcription. Finally, let's consider how hereditary diseases relate to our genetic makeup. Essentially, these diseases are caused by changes or mutations in our DNA sequence. Since our genetic code determines everything from physical traits to susceptibility to certain conditions, any alteration can lead to health issues. Understanding this connection highlights the importance of genetics in medicine and the potential for targeted therapies in the future. So, in summary, recognizing how DNA is organized within the nucleus, understanding the chemical properties of nucleotides, and appreciating the genetic basis of hereditary diseases all tie back to the fundamental theme of how our genetic information influences our biology and health.